Chapter 18. Food sensitivity as a digestive failure. Food sensitivity emerges when the digestive system fails to convert incoming food into forms that can be absorbed without extended interaction with the intestinal barrier. The reaction is not defined by the food itself, but by the condition in which that food reaches the epithelial surface. When digestion proceeds in sequence, proteins are reduced to small peptides and amino acids, carbohydrates to simple sugars, and fats to dispersed, transportable structures. These forms move across the membrane quickly, limiting exposure. When this sequence breaks, larger fragments persist in the lumen, increasing both the duration and complexity of contact with the barrier and shifting the system toward reactivity. The variability of food sensitivity reflects the variability of digestion. The same food can be tolerated under conditions where gastric processing, enzyme activity, and bile flow are aligned and can provoke symptoms when those conditions are disrupted. This inconsistency is a direct result of how substrates are presented to the intestine. When proteins are not fully unfolded and cleaved, larger fragments remain available to interact with the epithelial surface. When carbohydrates are not fully reduced, they persist and contribute to fermentation. When fats are not properly emulsified, they alter the physical environment and delay the coordinated processing of other nutrients. Each of these failures increases the likelihood of prolonged exposure. The sequence of digestion determines whether these fragments are cleared or retained. Gastric acid sets the structural state of proteins, pancreatic enzymes complete their reduction, and bile organizes lipids into forms that support efficient breakdown and absorption. When any part of the sequence is weakened or mistimed, substrates move forward in incomplete forms. These forms do not engage transport systems effectively, so they remain within the lumen and increase contact with the epithelial surface. The response that follows is driven by this exposure rather than by an inherent property of the food. Microbial activity amplifies this effect by acting on the same incomplete substrates. As fermentation increases, microbial byproducts accumulate alongside dietary fragments, altering the luminal environment and increasing signaling at the epithelial interface. This combined exposure changes how the intestine responds to subsequent inputs, reinforcing a pattern where sensitivity becomes more likely as digestion remains misaligned. Motility and timing contribute to the persistence of this state. Irregular gastric emptying and altered intestinal transit change how long substrates remain in contact with the epithelial surface and where that contact occurs. Prolonged retention increases exposure, while rapid transit can deliver partially processed material into regions not prepared to handle it. Both conditions reflect upstream disruption and contribute to variability in response. Food sensitivity, therefore, represents a failure of the digestive sequence to produce absorbable forms and clear them efficiently. It arises when substrates remain in the lumen long enough and in forms complex enough to alter interaction with the intestinal barrier. The specific foods associated with symptoms reflect the conditions under which they are processed rather than fixed properties of those foods. Restoring sequence reduces exposure, allowing the same foods to be handled without triggering the responses that define sensitivity. Immune response to poor processing. The immune response in the gut increases when digestion fails to convert food into forms that can be rapidly transported across the epithelial barrier. The system is built to encounter defined molecular units that pass through the membrane with minimal interaction. When proteins remain as larger fragments, when carbohydrates are not fully reduced, or when lipids are not properly dispersed, these substrates persist in the lumen and extend their contact with the epithelial surface. The immune response reflects this prolonged and altered exposure rather than the presence of the food itself. The structure of the substrate determines how the barrier interprets it. Small molecules move through transport systems with limited engagement, while larger fragments remain at the interface between the lumen and the membrane. This persistence increases the probability of interaction with epithelial cells and the signaling pathways they control. The response is driven by the form and duration of contact. As the size and complexity of the substrate increase, so does the level of signaling generated at the barrier. Microbial metabolism amplifies this process by acting on the same incompletely digested material. As microbes break down these substrates, they produce additional compounds that accumulate alongside the original fragments. The epithelial surface is then exposed to a combined environment of dietary and microbial products. This mixture changes the chemical conditions within the lumen and increases the complexity of signals encountered by the barrier. The immune response reflects the total environment rather than a single source. Timing and motility shape how this interaction unfolds. When digestion is aligned, substrates are cleared quickly, and exposure remains brief. When processing is incomplete, fragments remain in contact with the epithelial surface for longer periods. Irregular motility can further increase exposure by prolonging retention or by delivering partially processed material into regions where it would not normally appear. These changes alter both the duration and location of interaction, influencing the level of immune signaling. The location of exposure is critical because different regions of the intestine are adapted to different levels of substrate and microbial contact. When digestion is functioning properly, most interaction with complex substrates occurs distally. When is not, these substrates appear earlier in the intestine, increasing immune activity in regions that are not designed to handle sustained exposure. This shift amplifies the response and contributes to variability in how the system reacts to incoming food. The immune response to poor processing, therefore, reflects a mismatch between substrate form and the conditions required for efficient absorption. It arises when digestion fails to complete its sequence, leaving larger and more complex material in contact with the epithelial surface. The resulting signaling is a direct consequence of that exposure, shaped by the structure, quantity, and distribution of substrates within the intestine. Loss of tolerance as system instability. Loss of tolerance develops when the digestive system stops producing consistent substrate conditions and begins delivering variable inputs to the intestinal barrier. Tolerance depends on predictability. When proteins are reduced to the same small forms, when carbohydrates arrive as simple sugars, and when lipids are dispersed and matched with bile and enzymes, the epithelial surface encounters a stable pattern of substrates. Under these conditions, transport proceeds quickly and interaction remains brief. When this consistency breaks, the system is exposed to a shifting range of fragment sizes, concentrations, and delivery rates, and tolerance narrows as a result. Instability begins with disruption in sequence. Variations in gastric acid output, enzyme activity, bowel flow, and motility change how food is processed from one meal to the next. The same food can be reduced efficiently under one set of conditions and remain partially intact under another. This inconsistency alters the form of the substrate presented to the intestine. Larger fragments persist during periods of reduced processing, increasing exposure to the epithelial surface and amplifying signaling. Over time, this repeated variability changes how the system responds, making it more likely to react even when conditions temporarily improve. The distribution of substrates contributes to this instability. When digestion is aligned, nutrients are delivered and absorbed in a steady flow. When it is not, substrates arrive in uneven pulses or remain in the lumen longer than expected. These fluctuations create alternating periods of low and high exposure, preventing the system from maintaining a stable interaction pattern. The barrier adapts to this variability by increasing sensitivity to changes in substrate presence, further reducing tolerance. Microbial activity reinforces this pattern. As incomplete substrates reach the distal intestine, fermentation increases and generates additional byproducts that alter the luminal environment. These changes feed back into the system, affecting motility and the conditions under which subsequent meals are processed. The result is a cycle where variability in digestion leads to variability in microbial output, which then increases variability in the digestive environment itself. The immune response tracks this instability. As the epithelial surface encounters a wider range of substrate forms and microbial byproducts, signaling becomes less predictable and more reactive. The system no longer operates within a narrow range of expected inputs. Instead, it responds to a fluctuating environment where the same food may produce different substrate conditions depending on the state of digestion at that time. Loss of tolerance, therefore, reflects a system that has lost its ability to maintain consistent processing and delivery of nutrients. Is not tied to a specific food but to the variability in how that food is handled. When the digestive sequence is restored and substrate delivery becomes consistent again, the conditions that drive instability are reduced and tolerance can return as the system reestablishes predictable interaction with incoming nutrients.